In this activity, we're going to look at cells from tomato fruit pulp. This is a great example of how plant cells can be specialized for something other than photosynthesis or storage. In fruit tissue, we often see pigment organelles that help the plant attract animals to eat the fruit and disperse the seeds. As you observe these cells, pay attention to what stands out most, because it won't be chloroplasts this time. Tomatoes develop bright red and orange colors because of pigments stored in special organelles called chromoplasts. Chromoplasts are a type of plastid that store pigments other than chlorophyll. In tomatoes, the main red pigment is lycopene, a type of carotenoid, and it builds up as the fruit ripens. These pigments are not just for show, they play an important role in the plant's reproductive success by attracting animals for seed dispersal. Before we look at the images, pause and answer this question in your lab notebook. Think about what happens as fruit ripens and what plants gain by becoming brightly colored. I'm not looking for a perfect answer here, I'm looking for your reasoning. Go ahead and pause now, then come back when you're ready. Here's what tomato pulp looks like under low power. At this magnification, focus on the shape and size of the cells. This is under the lowest power. They are huge compared to others we've seen. To create this slide, the tissue is scraped with a scalpel rather than sliced, which causes them to be spaced out. Now we have both the low and medium power images, where an individual tomato pulp cell is easier to examine. As you look at these images, start asking yourself, where's the outer boundary of each cell? Can you trace the cell wall? Do you see small colored structures inside the cell? What color are they? Are the pigment structures clustered in one area or scattered throughout? Does the cell look like it has a large open space, or does it look filled in? At this point, resist the urge to label everything right away. Just observe carefully and think about what each structure might be. Now let's confirm what we're seeing. The cell wall is the rigid outer boundary that gives the cell its shape and structure. The small red or orange structures inside the cells are chromoplasts. These are pigment storing plastids. You can't see the membrane for the chromoplasts, so you're actually seeing clusters of carotenoid crystals. In tomatoes, these chromoplasts contain lycopene, which is responsible for the red color that increases as the fruit ripens. You may notice that chromoplasts sometimes appear clustered, often around where the nucleus would be, but they can also be scattered throughout the cytoplasm. Either pattern is normal. Now it's your turn to complete the drawing for this activity. Draw one tomato pulp cell based on what you observed and label the cell wall and chromoplasts. Focus on showing the pigment bodies clearly. Those chromoplasts are the main feature in this activity. Pause the video here and resume when you're finished. Now compare tomato pulp cells to the other tissues you've observed. Do they resemble the onion epidermis with its neat brick-like pattern? Or do they look more like potato tuber cells, which are larger, more loosely arranged, and specialized for storage? Use evidence from your observations, especially cell shape, spacing, and the types of organelles you see. Pause the video to answer this question. This is a great place to connect the lab activities together. In Alodia, you observed chloroplasts, which are green and specialized for photosynthesis. In potato cells, you observed amelioplasts, which are starch-starring plastids that stained dark purple with iodine. And now in tomato pulp, you observed chromoplasts, which store pigments like lycopene and contribute to fruit color. Even though these are all plastids, they are specialized for very different functions depending on the tissue and the needs of the plant. Note how they all differ in color, appearance, and function in your lab notebook. Go ahead and pause the video now. The big takeaway from this activity is that plant cells are specialized based on the role of the tissue. Tomato fruit cells contain chromoplasts, which store pigments that help the plant attract animals for seed dispersal. This also reinforces the idea that plastids are not all the same, but they all store something. Chloroplasts, amelioplasts, and chromoplasts are all plastids, but they are specialized for different functions in different tissues. In this activity, we're going to examine the tomato epidermis, the thin outer skin of the fruit. This tissue is specialized for protection, but it also has an important role in communication between neighboring cells. The main structure we'll be looking for today is something called plasmodesmata, which are connections that run through cell walls and link adjacent plant cells. The epidermis is the outermost layer of cells on the tomato fruit. These cells form a protective barrier that helps reduce water loss, protect against pathogens, and manage how the fruit interacts with the environment. Because epidermal cells are at the surface, their structure is adapted for protection. So we'll pay close attention to cell walls and how these cells fit together. One of the unique features of plant cells is that even though they have rigid cell walls, they are not isolated from each other. Plant cells can communicate through tiny channels called plasmodesmata. These pass through the cell wall and connect the cytoplasm of neighboring cells. Through plasmodesmata, cells can exchange water, ions, sugars, and signaling molecules. Basically, they help the plant coordinate what's happening across tissues. In some cases, plasmodesmata are not evenly distributed. They can appear clustered in specific regions where the cell wall is thinner. These clusters are called primary pit fields. Depending on the microscope image quality, you may or may not clearly see this term in action, but it's helpful to know what you're looking at if you notice a concentrated area of small dots or strands along the wall. Before we look at the images, pause and answer this question in your lab notebook. Think about what the epidermis does and why it might be important for surface cells to share information or materials with the cells next to them. Go ahead and pause now, then come back when you're ready. Now we're looking at tomato epidermis under low and high power. As you look at this image, start asking yourself, where is the outer boundary of each cell? Are the cells tightly packed like tiles, or are there gaps between them? Do you see faint granules or light region inside the cell that could be cytoplasm? Along the cell walls, do you notice tiny dots or fine strands that seem to cross the cell between cells? At this point, resist the urge to label everything right away. Focus on careful observation first. Now let's confirm what you're seeing. The cell wall forms the boundary of each cell and provides structure and protection. The cytoplasm is the living material inside the cell wall. In these images, it may appear as a light region or faint granules. It may not look dramatic, but it's there. The plasmodesmata are the key feature in this activity. They appear as very fine dots or tiny strands that cross the cell wall and connect neighboring cells. They can be subtle, so if you don't see them immediately, that's normal. Look carefully along the wall where two cells meet and see if you notice small connection points. Now it's your turn to draw what you observed. Draw two adjacent tomato epidermal cells and label the cell wall, cytoplasm, and plasmodesmata. Your drawing does not need to be artistic. It needs to be clear, accurate, and labeled. If plasmodesmata are difficult to see in the image, do your best to represent them based on the area where cells connect and make a note where they are subtle. Pause the video here and resume when you're finished. Now compare these tomato epidermal cells to other cells you've observed. Do they look more like onion epidermis cells, which are also protective and tightly packed? Or do they resemble potato tuber cells, which are storage cells and tend to be larger and more loosely arranged? Use evidence from the images, shape, packing, and tissue function to support your reasoning. Pause the video to answer this question. Now think about the difference between the tomato epidermis and the tomato pulp cells you observed earlier. How does the cell wall appear in each tissue? Epidermal cells are part of a protective surface layer, so you may expect stronger or more clearly defined cell boundaries. Pulp cells are internal and are more involved in storage and ripening, so their structure can look different. Explain how these differences support the function of each tissue. Pause the video to answer this question. The tomato epidermis is a protective surface tissue, but plant cells still need to communicate and share materials. Plasmodesmata provide direct connections between cells, allowing water, ions, and signaling molecules to move between neighboring cells. This activity reinforces a major theme in plant biology. Even with rigid cell walls, plant tissues function as coordinated systems, not isolated cells.